I'm Mr. Worski here, and this is the second screencast of the cell cycle or mitosis series. This is the last slide that we were left off on. If you hadn't gotten the picture down by now might be a great time to do it. If you have any questions, please, you can review the video again. You can read your textbook or even ask me a question in class tomorrow. So anaphase is the next phase after metaphase. So the APC is the anaphase promoting complex. It's a protein that will digest the kinetochore, the structure that holding the two chromatids together. The kinetochore spindle fibers will shorten, and their sister chromatids will separate and go to the opposite. So the identical DNA strands separate and move to the opposite poles. So here's a picture. Oh, wow. You see now, here's the centrosome, aster spindle fibers. These should be polar spindle fibers that connect, and the kinetochore spindle fibers are connecting to the kinetochores of the chromatids. The chromatids are now being pulled to the opposite poles. Beautiful. So draw the picture. Hit pause if you need to. Telophase is the last phase of mitosis. It is the opposite of prophase. So the chromosomes decondense. That means they get longer and skinnier. The nucleolus reappears since the chromosomes are getting longer and skinnier. The chromatid loops are able to associate with each other again. The nuclear membrane will reform. The spindle fibers become disorganized. That means the cytoskeleton is starting to form, and the cytokinesis can now begin. Now, cytokinesis is the division of the cell. Now, here's a picture of telophase. We start to see cytokinesis. The spindle fibers are starting to break down. You notice that we have the nucleus here. The nucleolus is right there. Draw the picture of telophase, please. Beautiful. Hit pause if you need to. Cytokinesis is the division of the cytoplasm. So there's two types of cells, animals and plant cells, that divide. So how do they, and they divide differently. So how does each one go through cytokinesis? Well, the animal cell, there's an actin protein around the inner membrane at the metaphase plate. It's like a belt. Just think of a belt just lining the inside of the cell membrane, and all of a sudden this belt starts to contract and get smaller and smaller and smaller. So if you had a belt, like your belt's contracting, getting smaller and smaller and smaller, eventually the cell will split in two, like you would if you had a belt that you were making smaller, smaller, and smaller. That is how an animal cell goes through cytokinesis. So we have the microfilament of the actin there, it gets smaller, 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 and two cells divide. But the cleavage furrow here, boom, like that. Now, notice the plant cell can't do that because the plant cell has a plant cell wall surrounding it. So plant cells cannot have divide the same way. So how do the plant cells do it? Well, let's go over how plant cells divide. Here we go. So vacuoles form across the metaphase plate. The vacuole fuses and the cell splits. It's right here. Vacuoles are forming across, they fuse, and now the cell has divided. Pretty simple. So what controls the cell cycle? Well, we know that the checkpoints control the cell cycle. We know the checkpoints between that G1 that can go into either GO or continue with the cell cycle. The checkpoint between G2 and M makes sure that the DNA is okay after the DNA synthesis. And we know that metaphase, we need to make sure the chromosomes are lined up in order for the cell to complete the cell cycle. There are also the presence of certain chemicals, like CDK, cyclin-dependent kinase, MPF, which is the M-phase promoting factor, or APC, which is the anaphase promoting complex. All those things must be present in order for the cell cycle to continue. If they're not, the cell cycle does not continue. For example, CDK. If you do not have CDK, you stop in the checkpoint of G1. The cell cycle control system is like a built-in clock of things that have to happen at specific times in order. And if you don't have those systems going in order, then the cell cycle will stop. 4. The size is the most important thing for the cell. The cell has to be a specific size, so it has to grow a lot during G1 through the protein synthesis phase. If it does not grow a lot during G1, it will not enter the cell cycle, will not go through G1. Instead, it will be part of G0 or go into G0. Environmental conditions, which is number 5, like pH. Hormones. Hormones can increase the cell division sometimes, right? Local regulators, PDGF, platelet-derived growth factor, will actually increase cell division so that you can heal after a wound. So the platelets are the things that help blood clot. They can release PDGF when they get to the site of a wound, and when that happens, the cells can divide quicker to heal your wound. And the density of cells, which is density-dependent inhibition. Usually, if a cell is bonded or surrounded by other cells, it will not divide because the area is already dense enough. However, cancer cells, they have that removed. They do not have the density-dependent inhibition, and they will continue to divide over and over and over again. So get those cell cycle controls. If you need to listen to that again, just move that bar so you can hear my lovely voice one more time. Mitosis. So single-celled organisms, they go through asexual reproduction or binary fission with mitosis and cytokinesis. That's how they reproduce asexually. In multicell organisms like us, we increase our number of cells. We can also maintain the surface area to volume ratio. Since we have a large number of cells, this allows for cell specialization. And if our cells die or if they are damaged, we can replace them by the cell cycle. Sometimes, though, we have abnormal cell division, which is also known as cancer. Cancer is characterized by there are no controls as far as cell division or the cell cycle goes. There is no density-dependent inhibition. They are not regulated by cell size. They continue to divide and divide and divide. Normal cells, they divide about 20 to 50 times, and then the cell dies because the telomeres are too short. Henrietta Lacks had some cells removed from her cervix when she had cervical cancer. Henrietta was an African American female, and they kept those cancerous cells of hers alive since 1951, and she's died a long, long time ago. Right now, if you added together all the cells from all the different labs that came from her, there'd be 400 times her body mass. Why do they continue to divide? Because they have very stable telomeres. Whereas our cells, our telomeres get shorter every time they divide. The HeLa cells' telomeres are stable and they do not get shorter at all. So what's the first step to cancer? Is transformation, and that's usually caused by some type of mutation. And the immune system usually keeps an eye on cancer cells and kills them right away. But all you need is for one cell to escape your immune system and you can develop cancer. So in cancers, the cells divide very quickly. They are very small because they are not allowed to grow. There's a wrong number of chromosomes, so there are no checkpoints in some cancers. They divide so quickly that the chromosomes get pulled every which way, and oftentimes you will have problems with chromosomes, but it does not a problem with a cancer cell because they don't specialize. They're not specialized at all. Problems with getting oxygen, and we're going to talk more about that later, but the tumor in the cells in the middle of the tumor or the growth have a decreased oxygen supply. So there's some way that needs to be a way for those cells to get oxygen or they can die. And sometimes in tumors you have necrotic cells or tissue within the tumor. Other times the cells actually give oxygen to the cells that don't have enough. So it's crazy. It's crazy, like they share their oxygen. So a tumor is a growth of or a small clump of unspecialized cells. Would be cancer. Cancer cells are unspecialized, and all they do is interfere with the proper functioning of tissue. And a tumor can either be benign, which is good, a good tumor. If a tumor were to be good, it's easily removed, or it can be malignant, which is hard to remove. And a malignant tumor is usually one that has metastasized, which means that they have spread throughout different parts of your body. So that is the last slide in the cell cycle. We will talk more about cancer later. If you have questions, please feel free to check your book, to watch the video over again, or to ask me tomorrow in class. Please have a good night.